Let's take a look at the structure of a thin filament. This is a diagram of a sarcomere. Here's the boundary of this one sarcomere, and we can see the thin filaments in blue projecting in to the sarcomere. Let's take a closer look at the thin filament. This is a closer view of the thin filament, and you can see using the color coding that there are three proteins that make up the thin filament, that these blue bead-looking structures make up the central core of the thin filament, but then there's this orange strand and these yellow spheres. And so let's take a closer look at all of that, starting with the blue protein, because that's actin. Actin makes up the central core of the thin filament. They're deconstructing this to show us that there's two types of actin, two forms of actin. There's what's called G-actin, which is just globular actin, which is like the bead-like portion. But the thing is, the thin filament is made out of a strand of actin, and so what happens is the G-actin can form a linear strand, just like beads on a string, and we would call this strand F-actin or fibrous actin. And so the thin filament is made up of two strands of F-actin that are spiraling around each other. Taking a closer look at each molecule of actin, notice how it has that well in it, that indentation. That's a binding site for myosin. And so each one of these actin beads can bind to myosin. So let's take a look at another protein that makes up the thin filament. Taking a look at the protein that they're drawing in orange. This is tropomyosin. It looks kind of like a strand that's spiraling around the actin core. And when you take a look at how it's spiraling around, it's very strategically located. And so remember we said that on each actin bead that there's a binding site for myosin, and look at how tropomyosin is covering that binding site up. So in other words, on our little diagram over here, a more simple way to show this is tropomyosin is lying right over the myosin binding sites on actin. In other words, tropomyosin is a regulatory protein. When it's in place, then it's going to prevent myosin from binding to actin. There has to be a way to move it out of the way to let myosin bind to actin. So tropomyosin, when the cell is at rest, spirals around the actin core, strategically hiding the myosin binding sites. And then the third protein that we see making up the thin filament is called troponin. And troponin, it's these little globular proteins. There's always three little globs. Two of them just anchor it in place. So one binds to the tropomyosin, one binds to the actin. So that's what I mean. It's like glue. But then the third component of troponin is a calcium receptor. Calcium is going to play a regulatory role in this too. In other words, I bet if calcium was available and calcium binds to troponin, that's going to be the trigger that's going to move tropomyosin out of the way, exposing the myosin binding sites on the actin core. Let me show you what I mean. Notice that tropomyosin, colored dark purple here, is strategically covering the myosin binding sites on actin. Calcium is becoming available. Calcium binds to the troponin. That moves tropomyosin out of the way, exposing the binding site. Myosin, the thick filament, binds to actin. When calcium is removed, tropomyosin goes back in the way, preventing myosin from binding to actin.